All right, so let's start first with consideration as to why we would like to do single-cell proteomics. And one very obvious reason to do single-cell proteomics is to be able to analyze cells of different types that are functionally different. For example, if we analyze a human tumor, we would like to know whether the proteins that we measure come from the lymphocytes or from the tumor cells. But there's also another more subtle reason, not less important, that even if we analyze cells from the same type, they might differ very significantly from each other. One very ostensible reason for those differences could be the phase of the cell division cycle, but there are many other reasons which might be less ostensible, but they're not less important. For example, differences between cells from the same type might exist because of different metabolic states, which lead to drug resistance. There can be differences in the degree of functional polarization of macrophage cells. And these two reasons are going to be the focus of what I'll share with you today, of my presentation. But there are many other additional reasons which can include senescence. Cells from the same type might differ because of the degree of cellular senescence. They might differ because of the degree of exhaustion of immune cells. So all of these factors contribute to important functional differences within the same cell type, not different cell types. And one of the sure signs of progress in the field, in my mind, is the fact that our fifth meeting, we have a number of presentations where we are able to analyze those differences within a cell type, not just compare two different cell lines, but be able to quantify and biologically interpret differences within the same cell type, which of course requires a high degree of experimental consistency in the measurements and higher accuracy. Another type of analysis that can be performed to infer, at least to some degree, these levels of specialization within a cell type, of course, is by single-cell RNA sequencing, which is very useful and widely employed. But it's also limited in the scope of what it can detect. For example, proteins such as p53 that are regulated primarily by protein degradation are poorly analyzed simply by measuring their corresponding transcripts. And this is true for many other proteins across the human proteome. And then it's not just protein abundance. I think what we really would like to measure are the various modifications of proteins that reflect their activities, such as phosphorylation or cleavage, localization, protein interactions, and so on. And being able to do these measurements and being able to do them very accurately holds the potential that we might start inferring signaling networks, information transduction happening within protein networks in the cells. And what for me personally has always been very exciting and has motivated our efforts is the ability to do this inference with minimal assumptions. Because if we measure the confounders and if we measure the proteins transducing the information, all we need to do is to condition on the confounders to be able to identify if the interaction is direct or indirect. So that's the aspiration, but let me review what is the technology to get there and then share with you some of the results from trying to do that. So as we try to measure smaller and smaller changes in protein abundance, differences within a cell type, now we need to be more consistent. We can tolerate to a smaller degree experimental variation. And sample preparation has been an essential milestone for us to complete before we can have these more reproducible measurements. And in this case, what I want to emphasize is not the miniaturized volumes. What's actually more important for us is the very high level of consistency that we can achieve in terms of protein extraction, protein digestion, degree of labeling. And this consistency is what begins to allow to do the measurements of much smaller protein fold changes. And of course, the ability to scale to prepare simultaneously thousands of single cells, because that allows us to reduce batches that otherwise undermine the quantitative nature of our results. The other thing that I would like to remind you from yesterday is the type of data that we get when we perform mass spectrometry analysis of single cells, of proteins. Proteins look like this, and they're peptides, like these peptide-like features. And the important message here that will be relevant to much that I have to say is that we detect tens of thousands of peptide-like features. And there's a challenge of how do we sequence all of these? How do we quantify all of them? So they're already in the instruments. Within the sensitivity of current commercial instruments that are widely available, there's this very high precision data, and we are interpreting only a small fraction of it. And that also brings me to the point that we discussed during the panel discussion session, that there are major opportunities of improvements just in terms of improving data interpretation. But if we're selecting one peptide feature at a time to analyze, isolate, fragment, sequence it, perhaps use isobaric mass tags to quantify, then we have limited number to analyze those features, because if there's 60,000 of those and each one being isolated requiring hundreds of milliseconds of time, then within 1 hour gradient, we can analyze only about 10,000. So most of the data, most of the peptide-like features are not going to be even touched from tandem mass spectra, and therefore we cannot reliably determine their sequence, and certainly cannot determine quantification using isobaric mass tags. So what can you do? One approach might be, if you're going to analyze only a subset of those features, let's choose that subset wisely. Let's make sure that every MS2 scan has a high probability of being identified. So normally only about 50% or 30% of our MS2 spectra have high confidence sequence assigned. We can try to push this to maybe 90% or maybe higher. And then we can pick the peptides from proteins of interest so that we increase the data completeness and we are able to address the biological questions that motivate the study. And one efficient way of doing that is to use real-time alignment that allows selecting precursors a bit more intelligently than the common heuristic known as top N. And just for the wider audience of people who are not familiar with the top N, when we perform an MS1 survey scan, we generally detect hundreds of peptide-like features. If we don't have time to analyze all of them, a common heuristic is to pick the N number most abundant ones, such as the 3 most abundant. And some of those might correspond to tryptic products. They're not interesting. We're not going to identify them. They might correspond to modified peptides. We're not going to identify them. On the other hand, by knowing the sequences that we're interested in and by knowing their corresponding retention time, we can choose the red ones. Maybe these are the ones that are going to help us answer our biological question. And because of technology to do the real-time alignment that has been developed, selecting peptides of interest has become quite feasible and applicable. But there is a challenge here that what if our list is very large? What if we have many thousands of peptides that we want to analyze, these red ones? How do we identify all of them? And there was this tension as we developed this project between me and Gray Huffman being the lead student behind it, where I wanted to push the throughput. I wanted to analyze as many peptides as we possibly can, and Gray was telling me correctly, wait a minute, if our list is very long, we're not going to have time for every peptide on our list. So some of them are not going to be identified. Our data completeness is not going to be maximal. And Gray kept pulling, I kept pulling, and he stood his ground. And then we found a synergistic solution that could satisfy both of us. And that synergistic solution is really simple. It's just prioritizing, choosing the set of peptides that are always going to get time, no matter what. We are going to have duty cycle time for them, and those that we are going to analyze if time allows. So let me explain this diagram a little bit. We perform an MS1 survey scan and we detect hundreds of precursors. And some of those are going to be the VIPs. They're going to get priority and they're going to be analyzed. And we do this in such a way that we ensure we're going to have duty cycle time. And others middle, bottom priority, they will be analyzed if time allows. So with this approach, we can operate at full duty cycle at all times, and still get the VAPs being consistently, reliably identified with the highest reliability possible. And that made Gray happy and made me happy. So it was a very fortuitous outcome of the pooling exercise. So when we implement this method on single cells, we see that we double the number of confidently identified peptides, doing everything the same samples, the same hardware. The only thing that is changed here is the logic that operates the instrument, and this is done with very widely accessible platform that everybody can utilize and start implementing. This approach also allows us to significantly increase data completeness, and this increase is most significant for challenging peptides, the peptides for which normally we would have the most missing data. Like in this case, only 30% data completeness. These are specifically chosen challenging cases. We double the data completeness for those, and the overall data completeness for the entire data set is also increased to about 74%. So this is a little bit about the technology that we developed, one approach to deal with the good problem that we have. Too many peptide-like features, not enough time. Let me tell you about a vignette of its application to one biological problem, in this case, studying primary macrophages derived from bone marrow. And these cells are considered quite homogeneous among immunologists. So when we started our collaboration with colleagues at Harvard Medical School with a group of Ivan Zanoni, his students and postdocs were really suspicious that we are going to be doing single cell measurements in these highly homogeneous systems. And they thought, you're wasting your time. You're not going to find anything interesting. And in a way, we analyzed both untreated and LPS-treated macrophages. And in a way, you can see their expectation being carried out with type 1 interferon signaling, because the proteins involved in type 1 interferon signaling have abundances that are quite homogeneous within a treatment condition. If you add LPS, pro-inflammatory stimulus, you increase their abundance. There isn't anything interesting. We didn't need to do single cell measurements. But now if you look at the phagosol maturation, you see a different picture. Actually, the variability is within a condition, not across conditions. And that's interesting, but of course we had these skeptical immunologists. They should have been skeptical, say, that's just some molecular variation. It's not functionally important. How do you even know it's real? Maybe these cells were stressed out or something. And that skepticism is very healthy because it's actually very testable. We could ask, are there functional consequences of this molecular gradient that we see within a treatment condition? And we measured about 30-fold dynamic range in phagocytic activity of those primary macrophages within a treatment group. This is not across the treatments, it's within a group. And if we look at some of the proteins that are significantly differentially abundant between cells with high and low phagocytic activity, we find some wonderful positive controls, such as the mannose receptor, that particularly made Gray happy. And then we can take these data correlating the protein abundance with functional phenotypes and color code our PCA plots and demonstrate that the initially discovered molecular heterogeneity within a condition actually correlates to the functional variation within these cell types. And furthermore, now we can ask the question, we identified some molecular and functional differences, but can we maybe measure something about how this is regulated? Not only describe its existence, but maybe get a little bit closer to understanding how are these differences within macrophages regulated. And we know that a primary regulatory signaling pathway within macrophages involves proteolysis. It's a very widespread regulatory mechanism. So the prioritized approach allowed Gray-Huffman to measure a number of proteolytic products, and we first validated the ability to do those measurements in bulk cultures using well-established methods. Gray found that the measurements in single cells agree very well with the bulk measurements, and then he could extend this analysis to single cells and find proteolytic products that are associated with either pro-inflammatory or anti-inflammatory panels of proteins. So switching gear here a little bit, I would like to look at the cell division cycle because that's a source of differences within any population of proliferating cells in culture. And we first did an experiment of identifying markers for the different cell division cycle phases. We specifically did not want to synchronize cells because it is very well established and known that synchronization is a major artifact and perturbation for cells. And the beautiful thing is that if you do single cell measurements, you don't actually need to synchronize cells in culture by adding inhibitors. Instead, we simply sorted cells based on DNA content, and we could identify proteins that are differentially abundant between cells in different phases of the cell division cycle. And in fact, for each phase, Andrew Leduc, in this case, the student who led those experiments, could identify not only one marker, but two markers, so that we actually multiple markers grouped into two phase markers. They're multiple protein markers, and we see that the correlations between markers for the same phase are positive and correlations between markers for different phases are negative. And if we do the low dimensionality projection of cells within that space, we can nicely separate them by cell division cycle phase, and that's just confirmation that the analysis works and gives us sensible results. Now, can we discover something new based on these data? Can we push our knowledge further? And one idea that we had for doing that was simply to correlate these cell phase cycle markers to all of the proteins that we measured and ask whether the correlations of a marker differ across the different cell cycle phases. So for example, here we find that proteins that function in polyubiquitination correlate with the G1, the S, and the G2 marker, but the magnitude of those correlations is different. So we can assign a statistical significance here and say that both in monocytes and in melanoma cells, protein polyubiquitination actually varies in a concerted, functionally enriched ways across the cell division cycle. And it's not only protein polyubiquitination, but there are a number of other Go terms that show such concerted variation across the phases of the cell division cycle that is similar between those two different cell types. And interestingly, there are also some that are different between the two cell types, and those involve proteins functioning in immunology, which in immune processes, which is perhaps not surprising considering that monocytes are immune cells and melanoma cells are not. Now, the melanoma cells that we study here are actually a widely used model system to study drug resistance, as reported in this beautiful article from Arjun Raju's group, where they use this system to study priming for drug resistance. And we wanted to see, can we identify this in our data? So when Andrew Leduc projected his data from melanoma cells, he saw those two clusters appearing, and Andrew being Andrew, he immediately suspected, there's going to be some technical artifact. What is this smaller cluster? And he went through and searched for correlation with all the various technical artifacts, and when he didn't find one, he just color-coded those cells with markers that were previously published, identified at the transcriptome level and published by the Arjun Raju's lab as being markers for priming for drug resistance. And we found that indeed the proteomes of these cells separate based on these markers. And furthermore, Andrew could identify a number of other functional groups of protein that differed significantly between clusters, such as proteins annotated to function in senescence, secretion, and so on. And furthermore, using the cell division cycle analysis, Andrew could identify that the cells that are expressed the markers primed for drug resistance, they spend more time in G1 phase, they divide more slowly. And beyond that, he could also identify molecular differences between their cell division cycles, how proteins go very differently with the cell division cycle in cells primed to become drug resistant versus those that are not. And Andrew noticed another thing. He noticed that if he correlated the total amount of protein in a melanoma cell to the cell size, there was a clear correlation. That's not very surprising. But you see that there are those two populations. The main cluster has the clear correlation, and the cluster B of the cells primed for drug resistance has a correlation, but they're clearly distinct, right? They separate. So why is that? Again, Andrew went to look for artifacts. Is this possibly correlated to a technical artifact? And when he didn't find anything, we started thinking, so what can this mean? So we have two clusters, and one of them essentially has higher concentration of protein per cell. Why could that be? Well, I spent a lot of my PhD work studying metabolism and cell division, and it's very well known. It's a classical result that when cells grow slowly, they accumulate reserve carbohydrates. Budding yeast accumulates trehalose. Mammalian cells, animal cells accumulate glycogen. So one hypothesis that immediately emerged is that, well, maybe these cells that are primed for drug resistance and grow more slowly accumulate glycogen. Let's see if we can find more evidence for that. So Andrew looked at the abundance of glycolytic enzymes, and he found that there is a significant difference of those enzymes being more abundant in the proliferative cells, being less abundant in the primed cells. So that's consistent with our hypothesis, but doesn't really prove it. And would it be nice to be able to do a more direct measurement? So then Andrew again went and he now sorted cells from the two populations, and he directly measured glycogen using a different orthogonal measurement. In this case, it's an enzymatic assay, it's not mass spectrometry. And he directly found that these cells that are primed for drug resistance accumulate more glycogen. And that's really a beautiful example of how having a very consistent sample preparation that allows to associate cell size with amount of protein can meaningfully interpret a difference between the protein concentration when that is detected in the data. So switching gears again, I want to tell you for another approach for dealing with the good problem that we have with the very many precursors that are present in our data. And the obvious approach is to acquire all the data. I told you that there are tens of thousands of precursors. We cannot analyze all of them if we are isolating one precursor at a time. And of course, there's a well-established method of data-independent acquisition that, regardless of what the accumulation time for tandem mass spectra is, allows to collect data for all of the fragments. But this approach, when applied, is usually used to analyze one sample at a time rather than multiple samples. And we were very interested in being able to increase the number of samples, single cells that can be simultaneously analyzed. And the other aspect is data-independent acquisition allows you to collect the data from all the peptides, not necessarily interpret or quantify these data. And in fact, for the most of the time of the existence of data-independent acquisition, these data were very poorly interpreted. Even for label-free analysis, we couldn't get deep coverage. And now we're going to make the spectra even more complicated. So merely collecting the data is no guarantee that we can make sense of them. There's this whole another level of how do we also enhance peptide sequence identification and do this synergistically with the added multiplexing. And this is what inspired us. That was the hypothesis to begin with. Let me orient you here. So we can analyze either multiple samples in parallel as done with isobaric mass tags as we've initially implemented with single-cell proteomics. We can analyze multiple peptides in parallel as done with label-free DIA. And we could also try to multiplex both of them and potentially increase multiplicatively throughput. Potentially, it's not clear that we could do it, but that was the test, and we are going to test this hypothesis with n equals 3, and we are going to hope that we can push it to n equals much larger than 3. And we weren't interested in just identifying peptides and proteins. We wanted to have a way to rigorously benchmark, are we getting accurate data? Not repeatable, not reproducible. I'm interested here in quantitative accuracy. And the way to evaluate quantitative accuracy is to have a solid benchmark to know exactly what the expected ratios are. And Jason Derks generated such solid expectation by mixing the proteomes of different species and different cell types into known ratios, an experimental design that is very familiar with all of my mass spectrometry colleagues. And then these mixed samples were either analyzed with standard label-free DIA, as conventionally done in the field, or they were labeled with non-isobaric mass tags mixed together and analyzed with a single sample. So in this case, using 3 times less time. And what Jason found is that we see a very substantial increase in the overlap of the peptides and proteins for which we get consistently signal across all samples when we use the plexDIA format. So despite the fact that we spent 3 times less time, we actually see that there is a larger number of proteins that are consistently quantified across all 3 samples. And the more different the samples are, the more significant this benefit is. And initially, when Jason found these results, he found this on his own. And when he brought them up, I was a little bit taken aback because label-free DIA is known as being our standard for data completeness. Can we do better than our best standard for data completeness? Turned out we can. So I'm really quite happy to see this level of data completeness that is made possible with the plexDIA. And there are more details here that we can go into if you have questions. Now, it's not just about data points. We want quantitatively accurate data points. So the mixed proteomes allowed us to compare the measured ratios to mixing ratios. And we found that plexDIA achieves the same accuracy about as label-free DIA. And they afford the same ability to identify differentially abundant proteins with using 3 times less instrument time. And that's detailed in this publication here below. Now, this is a slide that I included specifically because of our discussion yesterday for matching between runs during Ryan Kelly's presentation and then during dinner discussion. It's an experimental design that we use to benchmark our match between runs application, and that I want to share with you as a way that I think is quite powerful to rigorously benchmark how well it works. So we had a sample that was composed of mixed yeast and E. coli proteins, and we had another sample that had only yeast proteins. And then these 2 samples are analyzed together. As part of plexDIA, we perform match between runs, and then we look for identified proteins, either yeast or E. coli. And in this sample that has both, we see both. In the sample that has only yeast, we see only yeast. And the reason why we think this is very powerful is because if this sample was completely empty left, there would be no ions. So match between runs doesn't really have a chance to be trapped. But if you put another complex proteome, there are plenty of ions that can be mistakenly matched, and that allows for a more rigorous evaluation. And this works very well when we implemented it both at the MS1 level and the MS2 level, matching between runs. Of course, we developed plexDIA with the intention of applying this to single cells, and I'll just give you a short vignette of that application. In this case, sharing with you results from 2 different experimental platforms separated by 1 decade of development time. And there's a lot that I can describe about this result, but there are really 2 messages that I want to focus on. The first one is that timsTOF SCP is performing really well with plexDIA, allowing to reproducibly and consistently identify many peptides on fairly short gradients. So in this case, we had 15 minutes of active gradient time. But the other message is plexDIA actually works beautifully on this old instrument from 2011. So the coverage is not as high, and if you have the ability to guess the best, if you have the resources to get the best instrument, you should absolutely get the best instrument. We are very excited about using timsTOF SCP with plexDIA. But it's not the only way of doing it. So I want to, particularly for the sake of our community, for what we could have done earlier, I want to delve into the data from the old instrument. Could we have started the field of single-cell proteomics a decade earlier? So let's look at data from the old instrument. So this is the extracted ion current for a number of different peptides. And one advantage that I want to highlight of plexDIA is that when their proteins differ by 100-fold between here a pancreatic adenocarcinoma cell and monocytes, we are able to still identify the corresponding peptides in the less abundant, in the cells where they're less abundant, because we have this perfect matching of retention time. They happen into the same scans. There are a lot of additional features that allow us to identify those peptides in a way that we could never have done with label-free DIA. And then, as I discussed yesterday, these data allows quantification at both MS1 and MS2 level. And because the biases that we are likely to have at MS1 and MS2 level, certainly biases from interference, are likely to be independent, when we see agreement in the quantification at the two levels, we can have very high degree of confidence in the data. And these data, again, that I'm showing here are from the old instrument. The data from timsTOF are even more beautiful. But again, I come back to my point. Could we have started doing high throughput, very quantitative single cell proteomics a decade earlier? This is how the data looks like from the old instrument, comparing the abundances measured for hundreds of proteins in single cells and in the corresponding bow cultures. And one thing that I'll finish highlighting, which is a particularly substantial strength of the timsTOF SCP system, is the very high level of data completeness that we see within a plexDIA. So these are data from the SCP system. Now we see 98% data completeness for proteins measured in the single cells using plexDIA, and fairly good data completeness across different sets at 75%. So we are quite happy with that. All of the data that we have generated in my group, we have shared via various websites, and not only the data, but the metadata. I'm delighted to see that colleagues in the community, such as Chris, are reanalyzing our data and helping us have more confidence in the results. If somebody applies a different set of methods and gets similar results, that's wonderful, that's very healthy. And we would be very happy to facilitate the efforts of any other groups who would like to reanalyze, understand the data, reuse them. Let us know, and we'll be happy to provide anything that you don't find already provided. And as a final note, I want to finish sharing with you my belief that what you're seeing at the moment is a very early harbinger of what the field is about to see. I think we are sitting at the nose of a rocket that is about ready to take off, both because we already have a lot of data that is collected with existing hardware that simply we can better interpret to increase the depth of our coverage, to start quantifying all sorts of protein modifications. And there are all sorts of exciting opportunities to further empower the hardware, such as improving separations, as Andrew spoke yesterday, improving the sensitivity of instruments, increasing the brightness of the ion sources, using various forms of ion mobility. That can further help. And the one thing that I'm particularly excited about is the opportunity of climbing this pyramid together. I think that there are a lot of synergistic benefits of developing more high quality, non-isobaric mass tags that can further enhance our ability to sequence peptides. We are still collecting precursors and fragments for many more peptides than we are actually confidently identifying. And I think we can increase our ability to identify those peptides, both by intelligently optimizing the mass tags and by improving our data analysis. And Georg, a student in my group, has already made progress in that direction. And I should say that Georg has a poster that he can share with you the details on that, and so does Gray and Jason and Andrew. So the work that I mentioned briefly today is so represented in more details upstairs. So if you want to learn more about it, visit their posters. These are the Andrew, who led the melanoma work, Gray Huffman, with whom we pulled the rope, and Jason Derks, who led the development of plexDIA. I'll stop here, and I'll be happy to answer questions.
right, thank you for a great talk. Any questions from the audience? There's one we have online as well, but.
So with DIA methods, spectral libraries are a useful addition to the analysis. What is your recommendation with the plexDIA framework, particularly as it scales up to higher plexicities in terms of, if that's a word, in terms of spectral library design? Do you make a single plex library and deeply characterize it, and then permute that across the mass space, or do you make a high?
That's a wonderful question, David. Thank you. The need for spectral libraries was a major burden for DIA methods, a major weakness, because before one could do DIA, you had to generate a project-specific library. And for me, this was a big turn down from doing data-independent acquisition analysis. Deep learning has fundamentally changed that. Now we see fantastic results, indistinguishable from empirically acquired deep library, simply by predicting those libraries in silico with deep learning. And the functionality for doing that is already present in a number of tools, such as Diane. So to your question, yes, you can just make the library for one non-isobaric mass tag for one channel, and then you can add the corresponding masses to all the fragments and precursors and scale this arbitrarily to n-plex. We have seen that fragmentation is very consistent across the different labels that we used, so this approach works very well. In short, in silico, one can easily generate the libraries that are needed, and that is very convenient. It's actually an area, mass spectrometry for the wider community is an area where deep learning has really delivered in some applications. I think it has more potential to deliver more. I think there are exciting opportunities to also use it to improve further data interpretation, but in terms of predicting peptide fragmentation, it works wonderful.
Okay.
Thank you for your presentation. I got a question about the cell size and the protein quantity you see. Do you think that we are reaching a point where we have to kind of normalize to be able to be more accurate in the variations we are seeing on the proteome? And if so, what are your suggestions for that? Do you plan to have housekeeping proteins and normalize like we can do on Western blot or whatever?
So I think the best normalization is going to depend very much on the specific case. There has been extensive discussion subject to numerous Nature Biotech and Nature Methods papers on how to normalize. And it highly depends on the question that one is asking. There are advantages to having spike-ins to which you normalize. You can normalize to cell size. You could normalize to protein concentration. And ultimately, my best suggestion in that case is use different methods, compare the results from those different methods, and report and interpret only what is consistent across those different methods. I think this is really the best we can do. Any kind of normalization, which generally goes for processing steps, data analysis steps, can have its assumptions and biases. We try to minimize those, but they're there. And I feel happiest when we do 5 different things and our results are observed with those 5 different methods, then I have confidence.
So normalizing on the size of the cell sounds like possible on round cells. But what if you have a rectangle shaped cell? You don't have the third dimension to evaluate what is really the volume of your cell. So
yeah, and I think normalizing to cell size is not necessarily all that useful, because that is going to introduce a major component of variance in the data that is not necessarily biologically interested. It's just telling you cell size. With those dissociated cells that we work, even if they're neurons, once we dissociate them, they become spherical if they don't have points of contact. But I think the most frequent type of normalization that is performed is at the level of protein concentration. Essentially, you can divide the measured protein RNA abundances by the median abundance in that cell, or the mean, making, if you have missing data, that becomes more complicated, that has to be taken into account. But conceptually, you normalize the abundance of each protein to the overall abundance of all measured proteins. And I think that's, for most cases, I'm inclined to saying that's likely to be the best method. Not in all cases. It's still good to check. It's good to have spike-ins. But usually, I think that's going to be the first method that I would try.
Thank you.
Thanks for a nice talk. So I'm really intrigued by your comparison with the QE. So how much do you think that performance that you get nowadays is due to the sample prep on the cell-in-one, for example, versus previous plate preps, and the fact that the cell-in-one has only been out for a couple of years?
I think that's not a huge factor. So we see similar quality of data using minimal sample prep. I say in terms of sensitivity. So one of the really, for me, the big advantage of nPOP, the reason why I like it, it's actually not miniaturization. It's not low sample loss, even though these things are true. For me, it's really consistency of sample prep. So that's very helpful. We achieve incredibly high level of protein digestion, which has been harder to do otherwise, and high consistency, and very high throughput. We can prep thousands of single cells. So that's where I see the real advantage of the newer sample prep. But if we use minimal POP, which is just sorting cells into multi-well plates, slicing them by freeze heat, we analyze them by plexDIA, we see very similar results. So that helps. There are also advances in data processing and analysis. So I fully take your comment to heart, and I recognize that in 2011, 10 years ago, our coverage probably would have been lower also because data analysis and interpretation wasn't where it currently is. But I think we could have started much earlier, and by now we would have made a lot of progress. We could have developed much of the technology and ideas, importantly ideas, that we have now earlier. If we had the courage, if we were bold to try and do something that many people thought was impossible.
But you still were bold in the end, so
yeah,
that's good. Maybe just a follow-up with plexDIA and kind of future perspectives, because of course you're now making the MS1s more complex and the MS2s, so that you have any idea where the limit in terms of multiplexing will end up being? It's a wonderful question, and I can give you a very solid answer with respect to single cells, which comes from our bulk analysis. With the bulk analysis, we are able to quantify over 200,000 precursors per sample. And if in single cells we're quantifying 20,000 per cell, that means that we clearly have the space for at least 10 plex. And with the 20,000, we don't actually see this reaching any kind of asymptote. We don't think this is the hard limit. So we can say with confidence that we can do at least 30 plex for the single cells, sort of have the evidence for that. And then beyond that, I can speculate and say that I think we can very likely go at least to 100 plex with the single cells. With the bulk, I'll be very happy if we can get to 10 plex and preserve the quantitative accuracy. And there, likely as one start pushing that limit, what's going to happen is that there will be some precursors that now are going to start being affected more by interference. And as long as we have a good quantitative framework, and I think that's very feasible, we are working on it and have really good quantitative results on that, preliminary results. So let's say you use 20 plex with bulk samples. It could be that 10 or 15% of the precursors are affected too much by interference, and to remove those because they're not as well quantified, but you still get the 20-fold increase in throughput, which might be a good trade-off depending on the interest. I certainly don't think that the 3-plex is in any way limiting, and there are all sorts of really interesting directions to be creative and extend that. For example, take advantage of the high speed of TOFs and make lots of MS2 scans, because if now you have narrow isolation windows, lots of MS2 scans, each MS2 spectrum is going to start being simpler and simpler and simpler because we have narrow isolation window. or one can also use ion mobility and come up with different solutions. I think once we start thinking about this problem, there is a lot of potential to further improve and increase the capacity for multiplexing.
And I guess resolution will start playing a role again as we
That's
go.
right. Resolution certainly helps, especially at the MS1 level quantitation. Resolution and quantitative and then accuracy of the detectors clearly plays a substantial role.
Thank you.
Do one over here, and then I'll go to the other side.
Okay, so Gabriel Mazzucchelli from the University of Liège in Belgium. Consistency is very important within the same studio, of course, but it's even more important between labs. To be able to use data from repository and use and apply deep learning or artificial intelligence. So what would be your recommendation to be able to qualitatively evaluate the data and the consistency between all the data from a repository? Do we have to use QCs? What are your thoughts?
So we worked on a community paper, a white paper resource, where we specifically discuss what should be the benchmarks for evaluating. And in short, we have lots of recommendations as to how to do the experiments to maximize the probability of success. And we also have even more discussion as to how do we evaluate and report the data. So in terms of evaluating it, I think it's very useful to have benchmarks for accuracy, not just of repeatability, because your point is taken to heart. It's key. It's very appropriate because within one lab, you can have perfect repeatability and be perfectly wrong every single time. You just consistently measure the wrong thing. And the hallmark of being perfectly wrong and being perfectly confused is just having those scatter plots of peptide intensities on a 6 orders of magnitude dynamic range. Look at this scatter plot, how beautiful it is, and tells me nothing other than that you have something that's repeatable. I think the way to really benchmark things and to compare between labs is to have standards of accuracy, which can be based on spiked in peptides. Standards could be based on mixed proteome species, can be based on having fluorescent standards proteins that are expressed in the cell so that we can measure them with fluorescence and with mass spectrometry. Another metric that I think can be quite useful is looking for internal consistency in the data, such as between MS1, MS2 level signal. Could be consistency with proteins between different peptides from the same protein, with a caveat that their proteoforms begin to play a role, but one can potentially do this in a smart way. So I think that's a really important question that you're raising that is going to become increasingly important as these workflows are adopted in labs. If you have thoughts, you and everybody else in this room and the broader community, I would welcome you to click on this link here and share your thoughts with us, and they will be incorporated in the subsequent generation, and you will become a co-author as we keep developing this resource. But yeah, it's time to start seriously thinking about benchmarking. And the thing that I feel most strongly about is that we have to benchmark accuracy in a way that is comparable between labs, not just repeatability within my lab, within my instrument, because that just doesn't mean much.
Thank you.
It looks good, but misleadingly good.
We'll do one more question, and then we'll move on to the next presentation.
It's a quick one, thank you. I'm curious about the dynamic range that is possible in separating cells in different cell cycle stage. So you showed really nicely that you can see M as G1, but of course there are major changes happening at anaphase. And so were you able to see a little bit of that as well?
In principle, we should be able to do it. It's a wonderful question. Thank you for that. So we would love to be able to interpret the dynamic trajectories, be able to separate the cell division cycle into 100 time bins, associate each cell with a time bin, and then have the trajectory of each protein across these time bins, which in principle is possible, and we are actively working on it. We haven't yet done it to our satisfaction, and that's why we haven't included it in the paper of my presentation. But I think that's a beautiful example of something that will be enabled by single cell measurements uniquely that you couldn't do without having the single cell measurements by doing that